Agriculture and Agri-Food Canada, and Parks Canada. Justine graduated in 2014 from Brandon University with a Bachelor of Science degree. In 2021, she completed her Master of Science degree at the U of M, looking at resistance gene rotation to manage blackleg in canola. I'll just touch on a few points around verticillium stripe in canola. It was first identified on a research farm south of Winnipeg in 2014. This was the first confirmation of the Verticillium longisporum in Canada. Since then, the pathogen has been located across Canada. The casual pathogen is found in production regions globally, with yield losses reported in Europe from the disease in canola. Verticillium longisporum is a soil-borne pathogen that is brassica specific. Yield loss estimates and management practices are still being determined. So again, as we've said earlier, this webinar is being recorded and will be added to the YouTube channel through SIA. So I'll turn it over to Justine to share her presentation. And please remember, we've muted you and on entrance. So if you have any questions, put them in the chat function. Justine and I will be monitoring as the webinar goes. So we will get to them as soon as we can. Thank you.
Awesome. Thanks, Patty, for the intro. Can you hear and see my slides?
Yes.
Okay, perfect. Just wanted to do one last check before I go into black screen here. And I will try to monitor the chat, but yeah, feel free to give me stop me halfway through if I'm whipping through too much information here. But yeah, thanks. Really look forward to chatting with this group. I know for Saskatchewan, this is a newer disease that we're starting to see more and more of. I'm based in Manitoba, so here in Manitoba, we really like our canola diseases. We seem to not have enough of them, so we develop new ones, and that's what we've really seen here with verticillium stripe. So to start things off, a lot of the graphics are my own images just through work with the Canola Council. I've got a few stolen from the U of M and a few other researchers, so I can at least kind of provide the background image to what we're looking at and what we're working with in some of the field situations and stories behind some of the images. Because this is a newer disease, it's good to just identify it and learn what to be looking for. So I am going to go through lots of different images, and that's really the stage that we're at, is understanding what to be looking for and how to properly diagnose it in the field and how to compare it to other canola diseases that we're seeing. So to start off, the inner pathologist in me has to start by introducing the terminology and how I'll be kind of addressing both the pathogen and then the disease itself. So Verticillium longisporum is the pathogen and it causes the disease verticillium stripe. So the pathogen itself, Verticillium longisporum, is a soil-borne fungi, and it causes vascular diseases within plants. So life cycle wise, it's got really low mutation rates. It produces through asexual spores. But when we know we're for sure working with Verticillium longisporum is when we see mycosporotia. When we see that, we know we're dealing with that particular pathogen, especially when we see it in canola. So the pathogen itself was first identified in Canada in 2014, south of Winnipeg. This was the first isolation of it, but not the first one globally. So this is a pathogen that is found around the globe, and especially in other canola producing regions, in particular in Europe. So we've identified it here, and then it's now looking at what it actually causes, what damage it causes in plants. So this is a brassica specific. We do see other verticillium species. If you're a sunflower grower, you'd be dealing with Verticillium dahlia. That's something we do see in other crops, but different species, different pathogen. So longisporum only affects brassica plants, so we're looking at canola and mustards, and then a few other weed species as well. weed species, not wheat, just to clarify. So it is very specific to brassicas. So the pathogen causes verticillium stripe, and the disease itself mainly focuses and causes damage within the stem. And by the stem, we're going to see half stem senescence. We see plants starting to lodge because of that weakened stem. We start to see necrosis, so the tissue starts dying within the stem. This pathogen that causes the disease only allows for one disease cycle per season. So you're not going to see multiple waves of this particular disease. You're going to see it typically at the end of the year into harvest, and actually even post-harvest is when you see it much more predominantly in a field. It does really well, and the disease really amplifies under hot, dry conditions. So that's why likely the last few years we've really seen it ramp up. We've been in a bit of a drought, and that's really creating that pressure within the field and those great conditions for that disease to get in there and cause damage within the stem. So just a little backgrounder, and I know we had captured some of this information in the intro. But it is a new disease, so there's a nice timeline here to kind of look through how it's progressed the last few years and what's happened in the back end. So first identification of it, 2014. In 2015, CFIA conducted a survey all across Canada and ended up finding this particular pathogen in 6 of the provinces. So all of the prairies were in that survey, and that's where they were able to identify it. In 2016, there was also a name change. So you'll see both terms thrown up between wilt and stripe, very similar, but we don't necessarily see the really severe wilting symptoms that they see over in Europe. And so that's where the name change was over to verticillium stripe, because we see the stem striping more so than a wilt on the plants. Kind of look back the last few years into 2018, moving up to now 2023. We now know what we're looking for. So the symptoms become more obvious. We know the signs of the disease, looking for mycosclerotia in the field. So we're able to see that. Right now, in previous years, and especially in I think it was 2021 when it was really hot and dry, lots and lots of reports across Manitoba that we were seeing yield loss because of this disease. Hot, dry years aren't really conclusive for other disease development. Something like sclerotinia doesn't really do well when it's dry, but we were seeing a lot of damage within canola fields, and a lot of that had come back to looking at verticillium. So to kind of further understand what's happening in the plant, I think it's really good to address the disease life cycle. You are going to be seeing different symptoms throughout the growing season, and other signs of the pathogen's presence. So it's good to understand that and how it works. This is an image that you can find online at the Canola Council of Canada's website. It's in the Canola Encyclopedia. There's a whole page for verticillium. But it's good to kind of walk through what happens and kind of put emphasis right at the very beginning in the intro of this that it's a soil-borne disease. So it's going to have kind of similar management practices to a disease like clubroot, where anytime that soil is moving, you're moving the inoculum source as well. So via equipment, the wind, water, just anywhere that soil is going, you're having those spores. So from the mycosclerotia. So the mycosclerotia will be in the soil. They will attack brassica roots, and that's how they're taken up into the plant. So they're going to be taken up into the root tissue and then move through the vascular system. So with that moisture and nutrients going up into the plant, the pathogen is going to spread. And as it moves up, it gets into the stem, and that's where it really causes its damage. Once it gets up into the stem of the plant, you're going to start to see the colonization, where that's where the mycosclerotia really start to develop and produce kind of that next season's inoculum. So they get in there, they cause that stem to become extremely fragile. And that's where you start to see, some stunting in plants because you're not allowing all of that moisture and nutrient uptake into them. You'll start to see that premature ripening and plant necrosis. So that's the kind of the key area to focus on. And it's one of the first, or one of the earlier times in the year where you can start to see the symptoms. And typically when you'd start to see verticillium is moving in kind of late August, early August if it's been a really hot July. But you're going to see it up in that stem when you kind of peel back that outer stem wall. A lot of times the plants are going to look just kind of beige discolored. There's going to be on one half of the stem, but it's when you peel back that outer stem wall, you'll start to see the development of the mycosclerotia. Mycosclerotia are extremely tiny. So depending on how well your eyesight is, you may or may not be able to see the individual mycosclerosias themselves. It may just look more so like a grayish hue. So a hand lens will help show it. And I've got a few images here to show that real big difference between verticillium and something like blackleg and what those spores would look like. Okay, so we'll head into the next slide here. So I'm going to just walk through a bunch of the different symptoms and what to be looking for, and then do some comparisons with other common diseases before I start to wander into all of our questions in the field and the unknown components. Now, when we're looking at verticillium stripe symptoms within a plant, you're going to see a lot of symptoms are very, very common to other stressors, environmental stressors, other diseases. So when we look at a whole plant, when you see dead plants in the field, that can be from a whole host of different reasons. But like I said, it is a symptom, and it is something you will see verticillium do, is take that entire plant. You'll start to see them die back. To me, verticillium plants, because they start to senesce early, they look very similar to plants that are also moisture stricken or drought, that have been in a really bad or a dry field year. So you'll see that early senescence, and you'll also see a little bit of stunting potentially. Stunting something that when working in a lab, you'll see those types of symptoms when you're more in a controlled environment. Out in the field, it's a little tougher to see stunting from a disease like verticillium, but it's still a possibility. As we look at leaves are an interesting one, and this is where the conversation about, is it a wilting disease, is it a stem striping disease, how's that come into play? In the greenhouse, plants that have been infected or in the lab or growth chambers, what happens is you see this half stem or half leaf senescence start to occur. This is something when canola plants are reaching full maturity, they start to lose leaves, and it's a tough thing to really see in the field. I know this year I tried to mark a few plants that were showing this to follow up to see if they had verticillium, and it's a tough thing to point out in the field. This should be maybe an early indicator, but it's something to follow up with and then see if there's the mycosclerotia developing. So then coming back through, okay, so leaves have fallen off, they're changing. That's all very normal for a canola plant to do, but it might be just happening earlier on in the season. The focus is then coming into the stems themselves, and this is where you see that dieback. You're going to see internal discoloration, and you're going to eventually see that stem start to turn gray, and it's turning gray based on the presence of the mycosclerotia there that are developed. One that's another area to start looking, and where I see early symptoms is when I'm out there looking for blackleg, I'm cutting into root tissue. You see this grayish discoloration across that entire root tissue itself, and you're able to find it lower down. So that's something too that I've been really kind of looking at and focusing on, is taking cuts low down. Is there that grayish discoloration? As I move up closer to the base of the plant, where I'd typically be assessing for blackleg, is that gray discoloration still there? Am I seeing more black predominant wedges, which would be something like blackleg? Training the eye to see the difference between the two. So here's just some visuals to kind of capture the previous slide and the verbal descriptions. But here's the half stem senescence, and this is where, to me, it looks very similar to drought, where you see those plants, they look like they haven't had a shot of rain. They've kind of shriveled up on the one side, or very dry, very fragile. But the key feature here with verticillium is it typically only happens when it starts on a half a stem. So you can see here in this bottom image, there's the kind of beigeish discoloration that's occurring, where the other half of that plant stem is still very green. So verticillium is going to move up there. So that means the inoculum, the pathogen itself has moved up into the vascular tissue there and started to cause some necrosis, from cell damage. So it starts off as this grayish hue or kind of beigeish hue, and you can see here in the plant or in the canopy, all of the different plants, and you can see this grayish hue or beigeish hue that's starting to occur here. So it's something that, if that was a mature plant that was just drying down naturally, you would see it all the way around. You wouldn't see it just on that half stem. And here's just kind of further up. So now you've got the branching of the plants. So same one, the bottom half here is now green, where the top half has got that beige discoloration. You can see now the branches on that half of the plant are also showing themselves to be gray. So here's the full plant just kind of backed off. You can see this half of the plant, the stems are still green and vibrant versus the other half. So this is one of the earlier signs you would see from a pretty severe verticillium infection. You can see up, because this side of the plant is starting to dry down quicker, you can see a little bit of shattering occurring. And that's, like I said, that's just a natural dry down thing. Overall, that plant on that half is not healthy and not thriving. It's not getting that moisture up there to those pods to hold on. So you have this premature senescence and eventual ripening that's causing pods to crack. So mild symptoms. So saying to me, these all look very drought-like. You've got this plant starting to dry down. If you're to break branches off, especially some of those side branches that are turning that beige discoloration, and if you were to peel back that outer stem wall, that's where you should see really see the true kind of sign of Verticillium longiform's presence, and that's the mycoscrotia. So very, very tiny grayish hue. And this plant kind of zoomed in is off of up here. So you've got plants from the same field. You can see how discolored they are versus green, healthy plants in that particular field. These images were from, I think, 2021, actually one of my neighbor's fields. So really dry, nice big field, but it was starting to go down in areas. So any of the areas that were lodging, this is what the plants were looking like in that side of the field. And this was early on. So to me, it was kind of too soon for plant maturity and even harvest. This was kind of mid-August. So very, very kind of aggressive symptoms that year. So this is the discoloration that I'm going to kind of really focus on and talk lots about, and especially when comparing it to blackleg. This is that grayish hue across the cross section. So you've got mycoscrotia kind of scattered throughout. Very, very light, very subtle, but it's across that entire cut. And same here, every plant is showing it. It's almost like a starburst pattern is how some people would describe it. Very faint, but as that plant starts to decay and mature, you can see the mycoscrotia start to really develop within that cross section. One of the other ones, right, where this pathogen has moved up through the soil into the roots, and here we're seeing what the damage can look like within the root tissue. So this top plant has got the outer root wall starting to slough off it, where a healthy canola plant, you pull it out of the ground, it's waxy, it's pretty strong, it's not, crumbling in your hand. And so that's what we're seeing here with the verticillium plants. And same, you can see that same grayish hue that we see in the cross-section cut, but that we also eventually end up seeing up in the stem tissue. So here it is just to kind of closer up. When plants and canola plants do die off, if you're to go pull plants today, through that layer of snow, you're going to see everything like this. This is just a natural decay type symptom. But when you're seeing it end of August into September, right, those plants should still have really healthy roots to them, and they shouldn't be showing symptoms like this. So the really key sign is finding mycoscrotia. If you can find mycoscrotia, right, that is the deciding factor between it being, verticillium versus sclerotinia, blackleg, alternaria, the whole host of other diseases that we work with. If you can find mycoscrotia up in the stem, it's a pretty safe bet that you're dealing with verticillium. So like I said, here's, you can see it just at the top here of this particular plant, there's a little greenish hue there. So that half stem senescence, where the side that has senesced is just loaded with Mycosgrotia. These plants, when you try to pull them up and out of the ground, typically shred in your hands. They become very, very fragile and soft. So if you were to try to pull them out, if that soil's tough at all, they're breaking off. And then I showed this image earlier, but right, it's deceiving from the outside. It can just look like maybe a sclerotinia plant that with that bleaching that's occurred, but you've got to kind of peel back that outer stem wall, especially earlier on, to reveal the Mycosgrotia. So just this is some late season. So here's as close as I could get using an iPhone and a little zoom lens just to show the tiny little speckling. It'd be very similar to kind of that of ground pepper. So very, very faint, but those are the Mycosgrotia growing. As the plants start to really decay, you see Mycosgrotia all over the stem. You start to eventually see it on that outer stem wall. You see it in the inside of the plants as well. So it really starts to grow and attack it all. Right now is actually probably one of the best times to go look for verticillium symptoms in a field. You're going to be able to see a lot of true signs and finding the Mycosgrotia. You know, this bottom image is plants that have been left out, you know, probably in October a few years ago that I pulled. So they're really, really fragile. And I make note of them because it'll be extremely tough to find them next spring. Because these plants have decayed so much and broken down so much, a lot of the signs of verticillium disappear in the spring because those plants have completely kind of disintegrated and been absorbed into the soil. So very different of that of a plant that has blackleg, where if you find the root tissue at all, it's hard, it's woody, and it stays out there for quite some time. So I'm just going to pause quickly. Don't have any questions right now. I am going to switch gears into, you know, what we know about this disease. It's, you know, kind of limited. I've captured a lot of the symptoms that we know now, and that's been a huge finding the last 5 or 6 years, right? We now know what we're working with at the field level, and we're able to identify it against other diseases. We still have a lot of unknowns, which I will get to those, but for right now, I'll talk a little bit about what else we know. And the key part here is just distinguishing. So distinguishing between different plant diseases. This was one of my first kind of field scenarios working with verticillium. This would have been back in like 2017, 18, in that range. I was actually trying to do blackleg ratings, looking at different major genes that were deployed, and a lot of my plants were showing this starburst pattern. And this really complicated matters because I was trying to rate blackleg, and I'm like, well, whatever I'm working with is not blackleg. Like there's something else going on here. And this particular field, I stopped by kind of every 3 or 4 days and really was able to watch the progression here of, you know, the symptoms that we saw early on for verticillium, and then later on how that ended up looking. And that was one of the key things with verticillium, was the symptoms come so late, you typically miss it on any sort of normal canola disease survey. We're not looking at plants when they're, you know, pretty well ready to go through the combine. And that's where we really see the distinguishing features of verticillium is much later on. But here's that, like I said, head-to-head comparison of verticillium with the starburst, that grayish brownish hue across the entire cross section, versus a disease like blackleg, where it's a very predominant black wedge. So here's just another comparison between these two diseases. We do see these diseases together in the field. So just to complicate matters, they will be there and present and even colonizing the same plants. But if you're dealing with a blackleg issue, right, we have a whole host of different management options out there and available. Where with verticillium, it's quite limited at this point in time. So that's why it's good to know. And like I said, if you've got blackleg, you can do something about it. So you can see here just the difference in size of the kind of the key distinguishing sign for each disease. So with the verticillium, you've got the Mycosgrotia, super, super tiny, versus the blackleg and their pycnidia or their pseudothecia. So the pycnidia are, I wouldn't call them quite the size of peppercorns, kind of like peppercorns cut in quarters. So still very, very tiny, where verticillium is going to be that ground pepper in the end. So quite a bit of a size difference there. And then here's just that cross-section cut. So this is cutting through where you typically cut for blackleg, so right at the base of the plant into the root tissue. But saying you can see the grayish hue across it all, and the distinguishing mycosclerotia starting to develop, versus just that internal blackening where we see little pockets of black wedges, which are blackleg.
Justine, we had a question come in about varieties. Are you finding there's more susceptible varieties than others?
Yeah, I will get to a slide on that and go through some of the kind of the genetic background there. So I will come back and address that question.
Perfect.
But yes, yeah. So and this is where like something like blackleg, you really do have varieties to change and alter. Verticillium, we're starting to learn more about variety selection and some inherent tolerance that's out there in the marketplace already. And that becomes very evident when you start walking small plot trials or nurseries. You can see plants that have gone down from disease pressure, may it be something like blackleg or clubroot or verticillium. So I won't go over, I've hammered that one out enough. Here's the same or same stem with two different diseases. So this one's kind of neat because you've had to peel off the outer stem wall, and that's kind of the key piece. Blackleg's going to colonize on the outside of that stem, where if you're to peel that back, underneath it is where you would actually see the verticillium start to grow. And that's what's happened in this particular plant. This is probably a late season blackleg, so Leptosphaeria bigelobosa moving in, saprophytic, end of the season, just kind of no real yield loss there, but causing some damage just hanging out on the plant. The one that I think we do misdiagnose early signs of verticillium with is with sclerotinia. And once again, from Manitoba, so I work with sclerotinia all the time, other than our last few drought years, see lots of it. And I know into northern Saskatchewan, it's starting to become more and more of an issue. Any sort of humidity, right? Something like sclerotinia takes off. And aggressive sclerotinia, if there hasn't been a foliar fungicide sprayed, will cause the entire plant to become bleached or beige. And depending on the conditions, you will have that white mold either continue to grow or stop growing and dry down. So when you're in surveying, a lot of the time the white mold is gone and it just leaves these bleached stems. Something like sclerotinia, if you were to crack that stem open, you're going to find the sclerotia body, so more of a hard kind of marble-like item within it, where the verticillium plants are going to be the opposite of that. You're not going to find it. You're going to find that grayish discoloration being the mycosclerotia. But you do have to peel it back, and you can see how similar these two plants look on the outside on those stems. They also shred very similar. They're very fragile. Growers that struggle with both diseases will comment that, you know, when they're going into harvest, the plants are just fragile and they wrap right around the reels. So they just kind of shred and make a mess is what really happens. So that's one to really kind of look and watch for, especially earlier on. If you're not able to find the sclerotia body or see white mold growing, you might be dealing with verticillium, not sclerotinia. And prior to the call, we were just talking about gray stem and some of the symptoms that we're seeing this year, and especially into that northeastern Saskatchewan. Gray stem's not a common disease. We don't talk. Sorry, it is a common disease, but we don't talk about it a lot because there's not much we can do about it, and it's a late season disease. So gray stem is a saprophytic. It comes in late in the year. When you have moisture at the end of the season, this is what ends up happening. It's really just an advancement for plant decay. It'll help dry those plants down and kill them off a little bit quicker. So we see a lot of gray stem. Gray stem is going to cause, like I said, a gray discoloration on the outside of the stem wall, and it's going to be all the way around the plant. You're going to see it start at the base, so at the root, and start to grow and move up the plant. So it ends up just causing that entire stem to go gray. There's not, like I said, it typically doesn't result in much for yield loss. This is usually when the crop's supposed to be coming off. Versus, like I said, that's something like Verticillium. This is late in the season, probably a good month afterwards, but you can see that graying has come through the stem wall this time. But once again, when you start to peel it back, you can find all of that mycoscrotia. Going back to gray stem, these plants stay quite hard and firm. So if you were to pull them out of the ground, they stay intact. They might be a little soft, but they don't shred and dry down like Verticillium plants do. So covered off, what to be looking for, the differences between other diseases, kind of training the eye. The one other thing we know is just roughly how severe the disease is, or how much of the disease we see. Right now, I'm just showing last year's provincial survey data. This year's results have actually just been summarized and they'll be presented this week. But last year, what we were seeing, really targeted survey in Manitoba, lots of fields looked at, but we were finding it in 38% of the fields surveyed in Manitoba. And I just kind of put some emphasis on the Manitoba results because that survey's been looking at verticillium and the trends of it since 2018. They've been recording it. And you can see, sclerotinia really dependent. The numbers are really dependent on the environment. Something like blackleg is typically always high. We find a couple of plants in every field out there, upwards or over 80% of the fields surveyed. And then verticillium, and you can see this trend the last few years. The more we know what to look for, the more we find it. But we've also been in that drought, those really hot, dry summer conditions or growing conditions. This year's survey results were a little less. And if I move a slide here, I might be able to get the actual number. But they were lower across Manitoba. I think more so kind of around that 29-ish range of field survey, or 29% of field surveyed were showing early symptoms for it. The one key thing with any of the surveys is that they are conducted prior to swath timing typically. So 60% seed color change, which is quite early for a disease like verticillium. So it's always interesting to look at this, if you were to do this survey a month later, would this number double or triple based on what we're seeing? So Saskatchewan Ministry of Ag actually did this survey last year where they did a late season or very targeted survey to look for verticillium along that Saskatchewan-Manitoba border. So the general survey that's done at 60% seed color change didn't find any symptoms of verticillium, where coming back about a month later, that's where they were able to really capture and see the symptoms of verticillium. So it wasn't a lot of fields, but it was fields in the RMs along that Saskatchewan-Manitoba border, where out of those particular fields that they surveyed, 64% of them were showing the disease. So it's something, like I said, that everyone's got a much better eye for and looking for. And the more we train people on it, the higher that this number is probably going to grow in both Manitoba and Saskatchewan. I believe this year's Saskatchewan survey was finding it in more of their fields in that early survey. So like I said, people just know what they're looking for, and we're going to see that grow. But it's neat to then look at that and compare it to other diseases. This year, because we had dry all summer long, and then the taps started to really turn on later on in the year. We had diseases like powdery mildew, which I know was a big issue in the northeast corner as well. Those kind of late season diseases that do well with moisture, we saw a lot of, and something like gray stem that I mentioned the slide before, that's another disease that does really well in those types of conditions. So I've captured what we know-ish, right? We know what to be looking for. We know roughly, where it is in Western Canada, but there's still a lot of unknowns. And right, we're coming on I guess almost 10 years of working with this disease right now. So we're starting to gain more information and more knowledge on it, but we still have some really big questions that are unknown. So some of the questions that I get lots is, or get asked about are, what are the conditions that allow that pathogen that causes verticillium stripe to thrive in? Like, what does it need in the field for it to really produce a bunch of inoculum to really spread and cause eventual, yield losses within our plants? what can then be managed or what can be done to manage the spread and the severity of the disease? What are the yield losses? Are we seeing different losses based on different sets of management practices or different hybrids that are being deployed? And that becomes kind of the biggest question in the end is, what types of resistant sources are out there? Do any exist? Are we just working with tolerance? So like I said, lots of big questions and answers like this just don't come overnight. There's been a lot of money invested into Verticillium through federal funding programs and through the provincial grower group organizations. Just hearing the voice of growers, understanding that this is becoming an issue and that they are struggling to manage it on farm. So because this disease has been found in other areas, there's also a lot of great work to pull from. And of course, the growing conditions in Germany are very different than what we see here in Western Canada, right? Different soil types, soil conditions, environmental conditions, much shorter growing season here in Canada. And that's one of the key pieces to kind of keep in the back of mind is, any work that's done in Europe, they have a much longer growing season. So what that tells me is the pathogen has a greater window of opportunity to cause damage and infection within plants there. But as for the environment, a lot of work and work that we are starting to find here in Canada as well is it needs warm soil temperatures to start off. And I wish I had more time in the day to look at these types of things, but looking at, the soil temperatures we see in the spring versus the symptoms and the severity of the disease we see eventually in the fall, see how those correlate. In Europe, they find that it thrives in those hot, dry conditions. And when I say hot and dry, it doesn't have to be super, super hot, just over 15 degrees. So as long as you have temperatures like that early on, you're going to allow that pathogen to really move within the soil and move up into plants. And where we see the plant move or the pathogen move within plants is around that 23 degrees. That's kind of like the optimal growing condition for it to, or growing temperature for it to spread within plants. So a lot of that work's been validated over in Europe, and we are starting to see similar results here in Canada. University of Manitoba had a dispersal study kind of looking at the direction and the distance that the Mycosclerotia would move, in the air, in the soil, looking at those types of questions, and then what conditions does the disease actually really thrive in. And like I said, we all know we've been in a bit of a drought here the last year, especially in the southern parts of the provinces. Those hot, dry conditions have really allowed a disease like Verticillium to spread. In Manitoba, I know we see it from corner to corner within the province. So it is widespread. There's definitely areas that see maybe greater severity of it or damage. Why that is, it'd be curious to kind of come back and look at some of these environmental trends and why we are seeing it like that. Just a field, you can see this was actually a field of a few plants have been pulled from, but you can see the areas starting to lodge and fall down versus greener areas in the front. So that's, like I said, kind of standard of a lot of different diseases. But when you look down and then look into the canopy, you can start to see the gray twisting of stems, and the stems are quite fragile. Crop damage. It's always great to put a number or a yield loss number or dollar value or yield loss number out there to really understand the severity of a crop disease. I know that's something that I've been pushing a few of my researcher friends, like we really need to get a better grasp on this so we can understand the impact to canola and to growers' bottom line. So over in Europe, once again, very different growing season, but they see wide swings in the losses, yield losses from disease. Some reports and some research show really high numbers. You'll see them kind of in that 30 to 50% yield loss range. Others have even a wider spread range. They're kind of like 10 to 50%. So there's a wide window there, and it really comes down to the environmental conditions. So when the conditions are right, that's where you can see those really high levels of severity within the plants. So it's work that we need to conduct here. We're obviously dealing with very different germplasm, different growing conditions. So we need that to understand. One of the key components that it's not necessarily an issue for farmers, but understanding the impact to the end product. So thankfully, what they've seen is, if plants are infected, the end product, so that end canola seed is still good in the end. You still have a really good oil profile, composition of your proteins, fatty acids, glucosinolates, all of those things that matter on a quality aspect are unaffected. So losses are inconsistent. Sometimes they're significant and other times they're not. It's extremely variable. And I like to kind of think of it as something like sclerotinia. You're going to see wildly different losses based on the environmental conditions. No precipitation in the season, you're not going to see much for sclerotinia damage. Verticillium is going to kind of work on the or very similar to that. But if there's minimal precipitation and really hot and dry, you're going to see the losses. So kind of a reversal in comparison to sclerotinia, but environmentally dependent. Some of the early work here in Canada really is just trying to look at rating plants, trying to develop severity rating scales, developing lab techniques so they can actually work with the pathogen. There's a lot of the behind the scenes that need to be established and methodology that have to be used first before you can move forward and actually getting good ratings. So some of the work out of the University of Alberta was doing just that, looking at different ways to inoculate plants so they could then eventually do plant disease severity ratings, yield loss ratings. And in one of their, inoculation methods, they were seeing quite high yield losses. So that's likely maybe way too aggressive or severe to what we would see then at the field level. But you can see in this particular example, and for their work, they were using a 0 to 4 scale. And you can see, obviously, the loss of pods that have occurred as the disease severity increases. So saying this is preliminary work out of the University of Manitoba. The project was focused on screening companies' germplasm to see if there was any sort of background tolerance or resistance to verticillium. So we've got different sources here. That's what the color bars are. At the bottom is just different companies' lines. So the company B or source B, submitted would be 6 different canola or germplasms or hybrids for them to screen and test. Very similar, a 0 to 4 scale was used. Our poor Westar variety or hybrid was used again to be kind of the indicator. It's the same hybrid that's used in blackleg ratings as well. But so it has no sort of resistance to verticillium, so it rates quite high on that score. Everything that they screened was below a 2. So does that mean it has resistance? Does that mean it has tolerance? Right? It becomes that fine line of, where do you rate it? And because the industry hasn't come up with any sort of standard yet, this becomes a bit of a gray zone. And I know companies are now starting to identify their hybrids' tolerance to the disease or their kind of background resistance. This is something that companies obviously feel confident enough to do within their own internal breeding programs. But like I said, the overall Canadian industry, there has no standard in place yet. So you're going to see very different scores be presented, different scales, different methodology behind that rating. So that's very kind of key to know and understand, especially with working with growers and providing or recommending hybrids, is that there isn't a lot of standardization behind that rating that's going out yet. Something that the industry's working on, but we're, like I said, not quite there yet. So I hate to end or wrap a presentation up on the grimmer side of things, but this is a new disease with very limited management options out there and available. So because of how it works and like I said, understanding that disease life cycle is there's no fungicide that can be applied right now. So there's no foliar or seed treatment that can go on and protect plants. Right, this pathogen is moving in the soil up through the roots into the plant. So as of right now, there's nothing on the market, no fungicide that would help slow that down. Your typical management items that they're kind of like the obligatory ones that you got or that have to go out there just on any sort of disease management, controlling volunteers, controlling brassica weeds. anytime you give a pathogen its host crop, it's going to fulfill its life cycle and produce more inoculum. So good weed control is important. One of the issues with Mycalus grotia is that they're going to stay and hang out in that soil for quite some time. So we don't know how long yet. We don't know how, if they die off, similar to that of clubroot spores, where you see them exponentially die off year after year. We don't have a good grasp of that. Given the spread and seeing, canola growers on a 1 in 4 rotation and still have really bad verticillium is an indicator to me that it's there and out there in the field and going to cause issues even if you are on a good rotation. It's going to be the environment that showcases how severe that disease will be in any given year. Eventually we will have resistant hybrids or tolerant hybrids, and we obviously are working with some right now. They're just maybe not identified as that yet. So that will be an option eventually. But like I said, we all know what happens when you overuse resistant tools. We start to burn through genetics, and using clubroot as a prime example of that, and how quickly we're able to overcome some of those resistant traits that are out there on the marketplace today. Length of rotation, I had addressed that one already. And then biosecurity. And when we first started working with verticillium, that's a conversation that had come up. We want to minimize the spread of the disease. One of the biggest issues with verticillium is at harvest, we cut through the inoculum source. So we cut through the stem of the plant where all of that Mycalus grotia is. And what happens is it just goes poof up into the environment. So now it's coated the combine, it's up into the air. So it's spreading like wildfire. So sanitation and some of those on-farm biosecurity type plans likely won't work that great in managing something like verticillium. And given it's 10 years since being identified, we see it all across Manitoba and well into Saskatchewan now. It continues to spread, and we're not slowing it down by any means. So, like I said, don't like to end on a real doom and gloom type summary, but options are limited. The big thing now is just identifying the difference between other plant diseases. So Canola Council's got a lot of great resources available online, and I definitely push those new research projects. So once the summaries become available, they go up onto the Canola Research Hub. Really good if you want to dive into the specifics and understand the ins and outs of how some of this work is being conducted. But if you want just the easy route, head over to the Canola Encyclopedia. It's got a verticillium stripe section that has all you need to know about this disease. And yeah, best thing is to just train your eye, know what to be looking for, know what to be identifying, and kind of getting out there and training the eye right now. And hopefully over time, there'll be something more we can do about it. And with that, hopefully that gives us a little bit of time for questions.
Excellent. Thank you, Justine. That was very informative. If anybody has any questions, can you please type them in the chat, meeting chat, so that Justine can answer them. Otherwise, if I don't get anything in the next minute or so, we will end the call. I just want to let the group know as well, we will be doing another webinar in November sometime on water quality with respect to livestock and spraying. So be on the lookout for that webinar invite. And we're trying to do a couple more activities, in-person one maybe in December sometime, and as well in January or early February. So if you have any ideas for any branch events, please feel free to reach out to one of the executive members, and we can look into that event as well. And I'm not seeing much for questions coming up. I guess Justine has also shared her email and phone number. So if you have something that you haven't been able to ask on the webinar, please feel free to reach out to her and ask, or use some of the resources she has suggested. Again, on behalf of the Northeast branch of SIA, I thank you, Justine, for doing this webinar. It was very informative and much appreciated. So I think we will end and give people back 8 minutes of their day, since there are no further questions.
Oh,
So
thanks,
thank you
Patty.
everyone for joining. Yeah, thank you.